So did you have anybody Monday get the 4878 on their first try?
Yes.
Yes? Okay. Two? Did you get any of the ones you were missing, the 7800 or?
7400? No.
It might be too big.
10,000, yeah.
Yeah.
Just too big to get in. Just
Okay, let's get started. Home stretch. So I understand if you went to the mentoring session last night, Austin put you through your paces, so hopefully that was helpful. Before I start just going through some review questions, are there questions about material you would like to ask? Anything that's particularly troubling you, not quite sure about, et cetera, et cetera. Okay, before we get going, let me go through a scenario that I think is probably one of the more difficult ones to wrap your head around, and it has to do with positive control of the black operand. Okay? So remember when we're talking about positive control, we're throwing in this additional element of an activator is going to be involved. That's not involved in the negative control scenarios that we talked about. So let's say we had some E. coli growing up in a flask of broth that had no sugar in it. No sugar of any kind. Okay? What is the status of the lac operon at that particular time? Is the operator blocked or unblocked? It is blocked. Why? Because we don't want to transcribe those genes that are involved in lactose breakdown if there's no lactose present. Why is the operator blocked? What makes an operator blocked? A repressor is attached to it, and that repressor is in its active form. What gene produces that repressor protein? The regulatory gene. Okay? So now we add lactose. Does that initiate transcription of those lactose breakdown genes? Mm-hmm. Why? Is the operator still blocked in the presence of lactose? Nope. Operator's unblocked. Why? Repressor protein is in the active or inactive state? Inactive state. Okay? Why? What has caused that repressor to turn from its active state that binds to the operator to the inactive state that won't? Lactose. Okay? Lactose binds to that repressor protein and changes its shape. So what role does lactose play? A co-repressor or an inducer? It is an inducer. Okay? New wrinkle, we add glucose to the media now. Given a choice, which sugar will the bacteria preferentially use, glucose or lactose? Glucose. Okay, but if lactose is present, that operator is unblocked. So why aren't we breaking down much lactose? What's happening to the rate of transcription of that operand? It's really slow. Really slow. Why? Because the appropriate activator protein isn't bound. What does that activator protein need to bind? It needs cyclic AMP attaching to it to be in its active form. But what's the relationship between glucose level and cyclic AMP levels? Glucose is high, cyclic AMP is low. Okay, so if we add glucose, cyclic AMP levels drop. Therefore, there's no cyclic AMP to attach to the activator protein, and therefore rate of transcription of that gene will be really, really slow. So basically, the bacteria are preferentially using the glucose. If they use up all the glucose, what happens? Cyclic AMP levels rise. Now that particular molecule binds to the activator. It binds to the promoter region, and it speeds up rate of transcription of that LAC operand. Okay? So that's the most complicated scenario. But again, for the exam, you really need to concentrate on the scenarios of, in an inducible operon, what is the normal status? Why is that status happening? Understanding what the repressor protein does, its interaction with the operator. It's better if you can understand the big concepts like that, and then if you're given some scenarios, you should be able to work through those. Okay? Any other questions about things that might be troubling, or do you want to do some questions? Okay?
Do we need to cover the, is it ubiquitin?
I just mentioned ubiquitin in one context. I think Austin talked about it a little bit more last night. What ubiquitin does is it targets messenger RNA for degradation. And we mentioned messenger RNA degradation as one mechanism you can use to regulate gene expression. And so if you understand that ubiquitin is kind of the signal that can be attached to messenger RNA that says, break down, you're good to go. Anything else? Okay, well, I'll try to kind of point out some things as we go along, but if you haven't responded to the first question that's up here, why don't you go ahead and do that? And I'm not going to wait forever, and so once we get it around 20 or 25, I'm just going to go ahead and move on to the next slide. Okay, money seems to be on E. Correct answer. Okay, all of those are post-transcriptional modifications of messenger RNA. So remember, these are things that are going to happen after that original transcription event takes place, and then we start modifying that molecule to turn it into a messenger RNA. Okay? Alternative splicing is the function of these particular introns, and so that is why 3 is the correct answer and not 1. Is it found in all types of RNA? Do you find introns in transfer RNA? Ribosomal RNA? No. These are in the genes that code for messages. Coding segments of a stretch of eukaryotic DNA are the exons. Introns, non-coding, exons, coding. These are the charging enzymes. These are the enzymes that have to hook up the proper amino acid with the proper transfer RNA. I know you're trying to write down answers. I'll send you an email of this with the correct answers checked off, so you don't have to try to write all this stuff down. Okay. So it's really important that the correct tRNA get joined up with the correct amino acid, so that when the anticodon of the tRNA binds to the codon of the message, the right amino acid is put into position. Okay? So the 5' cap and the poly A tail do a couple of things. They help to regulate the stability of messenger RNA and how it's degraded. They also have a role in helping to bind the messenger RNA to that small ribosomal subunit. Several of you put ta-ta box. Do you remember what ta-ta box is? That's in transcription, okay? That is a region of the promoter in eukaryotic cells that RNA polymerase recognizes. That would have been last exam material. Okay, so the first event is the binding of the small subunit to the message. Then what happens? We get base pairing B of the activated tRNA to the messenger RNA. Then the larger ribosomal subunit attaches. Then we get covalent bonding between the first two amino acids. And then we're out of initiation and we're into the elongation phase. Okay? So the sites, if you're looking at the ribosome, from right to left are A site, B site, E site. Okay? So the tRNA that was in the A moves into the P. Okay. So when did we talk about this? We talked about this after we talked about the process of translation. And then we said, okay, when you make a protein,
There can be different fates for a protein. Some remain in the cytoplasm, some are secreted. The information about whether that protein is secreted is actually part of the gene, which is transcribed into a message, and that is the signal sequence. So you might want to look back at that slide again if you missed this question, which several people did, to understand what that signal sequence is doing. Basically, it allows that message to hook up to a ER membrane, and then the protein that's secreted goes into the ER, and then it can be transported out of the cell. What this particular question is addressing is that concept of a frame shift mutation. And remember that a frame shift mutation doesn't involve just a substitution of a nucleotide, but involves an addition or a deletion of a nucleotide. So why is it worse to have it happen at the beginning of a gene as opposed to choice A at the end of a gene? Yeah, because it affects everything from that point on then when you have a addition or deletion. So that frame shift affects the whole rest of the message. So it starts at the beginning. That's going to affect more of the message than if it happens at the end. Why isn't a codon deletion as bad? How many base pairs is a codon? 3, right? So how many amino acids would you change by that one deletion? You only change 1. Okay, everything else would be normal. Okay? What about a substitution in the last base of a codon? What effect might that have? Might not have any effect, right? Because remember the redundancy of the genetic code, and that redundancy occurs in that third position, typically. Okay, so this is nonsense mutation. Why not induced mutations? Could this be an induced mutation? It could be. It doesn't have to be. It could also be a spontaneous mutation. So while that's an answer that could apply, that certainly isn't the best answer. Okay. Remember the scenario. We said that most carcinogens are also mutagens. And so one quick and dirty way to test compounds for being carcinogens is, are they mutagens? And the Ames test uses bacteria on a plate. And by looking at the way they grow, we can determine whether they've been mutated or not. Okay, so what causes a frame shift? An addition or a deletion of one or more nucleotides. So the cat ate, E is missing, the rat. So what does that effectively do? From every point past this deletion, the rest of this message is going to be frame shifted. Okay? So that's the only one that's a frame shift. The other ones just involve kind of rearranging the DNA, but are not additions or deletions. One of them is a base pair substitution. When we started out this whole scenario of talking about regulation of gene expression in prokaryotes, I really tried to stress that bacteria are constantly being exposed to different environments. I used your gut as an example. Okay? So these ability to turn on and turn off genes as food resources change, for example, gives the organism an advantage. Why? Because you don't have to express genes you don't need, because that allows you to save energy. Now, this has the environmental aspect, but do these changes in the environment change the genome? Do they change the A, T's, G's, and C's? No. They change the way that those genes are expressed. Whoops. Hold on. Shoot. My check mark was wrong. So, which of the following when taken by the cell binds to the repressor so that the repressor is no longer able to bind to the operator? I'll change my check mark. Should be inducer. Okay? Co-repressor binds to that repressor protein and turns it into its active form, so it then blocks the operator. So this one should be inducer. This is the lac operant scenario. So I'll change that before I send it out to you. Okay, for the inducible operon, like the lac operon, normal status is blocked, no transcription. But when you add the inducer, then that starts the pathway's transcription. So the presence of the lactose is what turns on transcription of that operon. Okay, so this is the inducer. When would this be the correct answer? Not if it was a repressor protein, but instead if that were an activator protein, like the CAP protein. Cyclic AMP reacts with CAP and changes its conformation. But this is asking about the repressor protein, so the correct answer there is A. Whoops. That's why I took that out. This has the control of several. So the important relationship here is the relationship between glucose and cyclic AMP. Because if this is going to increase and bind, we've got to have cyclic AMP binding to it. So we need a decrease in glucose to get that increase in cyclic AMP. If there's no tryptophan in the media, they need to make that amino acid. It's not available from their environment. To make the amino acid, those genes have got to be transcribed. To do that, the repressor has to be inactive so the operator isn't blocked. This choice would stop transcription. We don't want to stop transcription of the tryptophan synthesizing genes when there's no tryptophan in the media. Regulatory gene is outside the operon. The operon consists of the promoter, operator, and the structural genes. The inducer is the molecule that determines whether the operon is blocked or unblocked, but it's not part of the operon. So the only one that is correct here is the promoter region. So knowing what makes up the operon is important. Euchromatin, loosened DNA structure, transcriptionally active. Heterochromatin, compacted, transcriptionally inactive. Methylation, bar bodies, transcriptionally inactive, highly compacted DNA, all lead to heterochromatin. Histone acetylation loosens the DNA structure, euchromatin. So, review your terminology about the state of chromatin. All of those cells have the same DNA. So they're going to have the same promoters, they're going to have distal control elements, they're going to have enhancers, they're going to have DNA bending proteins. The difference is that different cells have different protein activator molecules. Those activators interact with the distal control elements, cause DNA bending to occur, and then recruit other proteins to the promoter so that transcription can occur. You right? Yep. So this is the RNAi. Okay, this is a defense mechanism against viral infections. When the cell sees that suspicious-looking RNA molecule, the COP is activated, and the RNA is broken down. are these called? What genes have the normal function to suppress tumor growth? Those aren't proto-oncogenes, are they? Those are the tumor suppressor genes. Okay? Proto-oncogenes are normal versions of gene that code for cell growth factors. If those genes become mutated by a carcinogen, then the proto-oncogene becomes an oncogene. Okay? If a cell has a retrovirus infection, that can convert a proto-oncogene into an oncogene. Are these underexpressed in cancer cells? No, they become overexpressed in cancer cells. So as a result of mutation, that mutated proto-oncogene, an oncogene, is now expressed too much, and we get too much cell growth. Okay? Cancer cells wouldn't overexpress these tumor suppressor genes, they would underexpress them, produce too little. The reason these are protective is that they encode proteins that promote DNA repair. Sometimes they'll arrest the cell cycle temporarily so that the cell has a chance to repair. And then if the cell's too damaged, what happens? Programmed cell death is initiated. The cell sacrifices itself and dies. That RAS protein is one of the signaling proteins in that pathway that joins the binding of a growth factor to translating that into some kind of action. So a mutation in this RAS proto-oncogene can cause a hyperactive signaling to occur, where the signaling occurs even in the absence of that particular growth factor. I suppose some argument could be made that's probably not a good choice, because if the signal's received properly, it results in cell division. So if I'd ask this question on an exam, I'd probably have to go back and go, yeah, that's an acceptable answer too. This is the better answer. Got my arrow out of place again. PDGF, platelet-derived growth factor, is a growth factor. Okay? It's the one that binds to the receptor, and then if the proper signals are sent, the RAS protein, which is encoded by a proto-oncogene, continues relaying the signal for cell division to occur. So this is actually the growth factor. It's not a gene. So that's another one I've got to change. Why is P53 called the guardian of the cell? Because this is a tumor suppressor gene. And remember, products of that gene stimulate repair, stimulate apoptosis if needed, slow down the cell cycle to give time for repair to occur. What does BRCA stand for? That's one of the breast cancer genes. Okay, the normal version of this gene is a tumor suppressor gene, which does this. If you have a mutation in this gene, your odds of breast cancer are markedly increased because you're knocking out a tumor suppressor gene. It's not a proto-oncogene. Okay, it's a tumor suppressor gene. So, a fair amount of kind of terminology that you've got to make sure that you understand. A lot of kind of definitions almost. You can't answer these questions if you don't understand what these particular terms mean. So you might want to consider making some vocabulary lists so you understand heterochromatin, euchromatin, proto-oncogene, oncogene, tumor suppressor gene. You got to know what those things mean to be able to answer questions relative to them. So, any last minute questions? Dr. Martin.
Those of you who have lab today, please bring something, a computer or a smartphone that you can use to do